welcome to the next chapter of biology, which is genetics, variation, and heredity. So let's get started with this chapter. This is the last chapter of biology of the course, that is genetics and variation. So the topic of evolution, which have been a part of the syllabus, have been deleted for this academic year. So we will not be discussing the topic of evolution here. We will be just discussing about heredity and variation in this lecture. So let's get started with the basics of variation and heredity. First topic in this chapter is accumulation of variations during reproduction. In the previous chapter, we have discussed about reproduction and what is the significance of reproduction. There we have seen that when organisms reproduce, the offspring shows minor variations, and these variations between two organisms of the same species can be due to certain inaccuracies which is occurring during the process of DNA copying. However, these variations are very less in case of asexual reproduction, whereas it is more in the case of sexual reproduction, because in the process of sexual reproduction there is the process of fertilization where the exchange of genetic material happens between the paternal and the maternal chromosomes. Some of these variations are useful variations and they help the organism to adjust to the changes in the environment, whereas certain variations do not help the organism to adjust, and as a result of which those organisms will die, and as a result of which those species can become extinct. So some variations are useful, whereas some are harmful for the organism because it can result in the extinction of those species. So let us see one example which is given in NCERT regarding how variation is significant in a bacteria. So here you can see this is the first generation, this is the second generation, and this is the third generation. So in the first generation we have one bacteria, and in the second generation that first bacteria have given rise to two daughter bacteria. Now you can see that their basic body plan is the same, but you can see minute variations over here and over here, right? So there are very less variations, but their basic body makeup or the body design is same. Now this bacteria is going to give rise to another two bacteria, and again you can see some variation that is in the next generation. Again you can see some variations over there. Some of these variations, as you can see, are either seen in the parental generation or in the first generation, that is the first filial generation. So some variations like this pink color as well as this blue color in the middle circle, these are new characters which was not present in any of the parents, right? So certain variations can be inherited and certain can be by mutations, and we have discussed about mutations in the previous chapter. That is, it is sudden heritable changes which is happening in the gene sequence of the organism, so that results in certain phenotypes which were not present in the previous generations. So this is about variations and the need of variation. Variation is extremely important for the survival of the fittest organisms in the environment. Now let us study some basic terminologies of heredity and evolution. Heredity means it is the continuity of features from one generation to another generation. What is variation? Variation is the similarities among all individuals and that belonging to have a slight difference. That is the degree of difference between the parents and offspring. That is known as variation. And genetics is a science of heredity and variation. So heredity is about the transfer of characters from one generation to another, that is transfer of similarities between one generation to another generation, and variation is the degree of differences between one generation and the next generation, and genetics is the study of heredity as well as variation. Let us now talk about two important terms. One is known as genotype and another is known as phenotype. Genotype is the genetic constitution of an organism, whereas phenotype is the physical appearance of a trait. For example, if you are tall, then that tallness is known as the phenotype, whereas this tallness is due to a genetic constitution, and that genetic constitution that is resulting in a phenotype is known as a genotype. So that can either be a capital T, or it can be a capital T small t. If your genotype is small t, then that means you are going to be dwarf. If it is capital T, you are going to be extremely tall. If you are capital T small t, then you are moderately tall. But if you are small t, then you are going to be dwarf. So this is known as genotype. Now what are these letters? These letters are nothing but alleles. Alleles are the contrasting characters of a gene. Genes are present on chromosomes and they are the structural and functional unit of inheritance. Genes always express in a pair, and this is known as alleles. So two letters are used to represent the allele or the genotype, because one is coming from the parent father and other is coming from the mother. So that is why we are using two letters to represent alleles. One is from the father and other is from the mother. That is why we are using two letters. Never represent alleles as a single letter, because in our chromosome both maternal chromosomes are also there and paternal chromosomes are also there. So that is why we are using two letters for the representation of any alleles, and alleles are the alternating forms of a gene. Now let us talk about Mendel's experiment. Gregor Johann Mendel was known as the father of genetics. He was the first person who have dealt with various experiments in genetics, and his experimental object was a pea plant. And do you know what is the scientific name of pea plant? It is Pisum sativa. So this Pisum sativum was considered as the experimental object by this particular scientist, that is Gregor Johann Mendel. So he have calculated different varieties of pea plants. What are these variations in pea plants? So let us see that variations one by one. It can be variations in the seed form, in the cotyledon size, in the flower color, then in the pod form, pod color, stem place, or it can be in the stem size. So based on the seed form, either it can be round or it can be wrinkled. So this round and wrinkled are going to be its phenotypes. So what are going to be its genotype? If it is round, we will represent it by capital R, capital R. If it is wrinkled, we will represent it by small r, small r. I am just taking the pure condition, that is known as the homozygous condition. If both the alleles are capital or both the alleles are small, then it is known as homozygous condition, and if there is a capital or small r condition, it is known as heterozygous condition. So in the second case, you can see yellow seed and green seed. So yellow seed is more dominant than green seed. That is why you will put a capital Y over there, and here you will put a small y, small y. Next is the flower color. First one is white. So white you will put capital W, and violet is the recessive trait that is not so prominent in the population. So that is why it is small w, small w. Next is the pod form. If it is a full-sized form, then it is known as full, or it is inflated. Inflated is denoted by capital I, and constricted is denoted as small I. Green pod is denoted as capital G, and yellow pod is denoted by small g. Then axial and terminal pods. Axial pod means if it is present at the tip, if it is present like this. If this is the leaf and this is the stem, the angle between the stem and the leaf is known as the axil. So if the bud is present over here, this is known as the axial bud. Whereas if the bud is present at the tip, it is known as the terminal bud. And it is seen that majority of the pea plants is having bud in the axils. So axial pods are denoted by capital A, and terminal pods are denoted by small a. Next is regarding the stem size. If it is tall, then we will denote it by capital T, and if it is short, we denote it by small t. So this is the representation for the various genotypes of different characters which were selected by Mendel. So now he have carried out different types of crosses, that is the experiment in which crossing was done between pea plants of different characters. If the cross is carried out between two organisms which differ in a single character, then it is known as a monohybrid cross. Once again, if a cross is carried out between two organisms that differ in a single character, then it is known as a monohybrid cross. That means if I am taking two pea plants, one which is producing the round seeds and another which is producing wrinkled seeds, then the cross is known as a monohybrid cross. Whereas if I am taking two different organisms which differ in more than one character, let it be two characters, then it is known as dihybrid cross. For example, if I take one particular plant which produces round and yellow seeds, and I cross it with another plant which produces wrinkled and green seeds, then that cross is going to be a dihybrid cross. So now we are going to study how a monohybrid cross is carried out and also how a dihybrid cross is carried out, and based on that we will study the various laws of inheritance. So let's get started with the first cross, that is monohybrid cross. So this is the picture for monohybrid cross. This is well explained. So let us study with the help of this diagram. So here I am going to take two plants. One pea plant produces yellow seeds and another one produces green seeds. Since yellow is dominant, we will represent yellow as capital Y, and green is recessive, so it is denoted as small y, small y. So here you can see that this is capital Y, that's the dominant plant, and this is small y, that is the recessive plant. And both of these are in the homozygous condition. That means that produces only one kind of gamete. If it is homozygous condition, then it will produce only one type of gametes. If it is heterozygous, for example small capital Y, then it will be able to produce two types of gamete, one carrying capital Y and another carrying small y. If it is having something like this, capital R, capital Y, small y, then how many different types of gametes can be produced? First one is going to be capital R, capital Y. Second can be capital R, small y. Next can be small r, capital Y. And the next can be small y, small y. So this is how we are making the gametes. So first of all, if we have a trick for understanding how many gametes we can produce, then it is easy for us to write down the gametes. So for that we have a trick. What is the trick? The trick is given by number of gametes is equal to 2 raised to n. This is the trick, where n is the number of heterozygotes. If it is capital Y, both are homozygous. So the number of heterozygotes is 0. So we get only one type of gamete because 2 raised to 0 is 1. Whereas if I talk about capital R, capital Y, here we can see one heterozygote. So n is equal to 1. So it will produce two types of gametes. Now what are the two types of gamete? Capital R with capital Y and another is small r with capital Y. Next one is the case of capital R, capital Y, small y. So in this particular case, it is having two heterozygotes, so it will be able to produce 2 raised to 2, that is 4 different types of gametes. And what are those 4 different types of gametes? One is capital R, capital Y, capital R, small r, capital Y, and small r, small y. So these are the different types of gametes which can be produced from capital R, small r, capital Y, small y. So now let us talk about the monohybrid rows. So here, since it is capital Y, there is no heterozygotes, so only one type of gametes can be produced, that is capital Y. Here it is small y, so it is going to produce only one type of gamete, that is small y. Now these two gametes upon fertilization results in the formation of a diploid cell again, and that is going to be capital Y, small y. Now this capital Y, small y is going to be the first generation that is produced by the crossing of the parental generation, and this cross, that is for the formation of first filial generation, we have to carry out cross pollination of parents. In the next step, what are we going to do? In the next step, we are going to take selfing of the F1 progeny. Selfing means you are going to take two different flowers of the same plant and the crossing is carried out between two different flowers of the same plant. So that is known as selfing. Selfing of the F1 progeny. So you are going to take this capital Y, small y. It will be producing two gametes. One is capital Y and another is small y. Similarly, another flower is also producing capital Y and small y. Now you are going to carry out the cross. That is capital Y, small y with capital Y, small y. So first of all, you have to generate the gametes. So here the gametes is capital Y, small y. Here also the gametes is capital Y and small y. This capital Y and this capital Y results in the formation of capital Y, capital Y. These two will result in the formation of capital Y. These two will result in the formation of capital Y, small y. These two will again form capital Y, small y. And these two are going to form small y, small y. So these are the different types of offsprings which can be produced, the genotypes of the different groups of offsprings that can be produced. But better way of calculating or finding the different types of gametes is by using a Punnett square. Now what do you mean by a Punnett square? Punnett square is a technique which was developed by a British scientist named Reginald C. Punnett, who was a British scientist, and he had developed a technique for easily calculating the gametes. How to do, I will be telling. First of all, we know that for carrying out the selfing, we have two organisms, right? Or two different flowers. So the first one produces gametes, that is represented on one end of the square, and in the other side, that is over here, we are going to represent the other gametes. So now you have to carry out the cross like this. This y with this y is going to be capital Y, capital Y. This capital Y with this small y is going to be capital Y, small y. This small y with this capital Y is capital Y, small y. And this small y with this small y is small y, small y. So this is how you are generating a Punnett square. So here you can see that 1 is going to show capital Y, 2 are going to show capital Y, small y, and 1 is going to show small y, small y. So what is the genotypic ratio? Genotypic ratio is coming out to be 1 is to 2 is to 1. Here capital Y and capital Y, small y is going to be yellow seeded, whereas small y is going to produce green seeds. Because there is no dominant characters, only recessive characters are there, and as a result of which there is no expression of the superior character, as a result of which only the recessive character will be expressed over there. So that is why you are getting a small y, and that is representing the green seeds. So here the number of yellow seeds is going to be 3 and the number of green seeds is going to be 1. So this is going to be the phenotypic ratio of a monohybrid cross. So in general, for any of the monohybrid cross, the genotypic ratio is going to be 1 is to 2 is to 1, and the phenotypic ratio is going to be 3 is to 1. So this is the phenotypic ratio and this is going to be the genotypic ratio. These two you have to study, the phenotypic ratio as well as genotypic ratio. Now what is the significance of this phenotypic ratio and genotypic ratio? Suppose you got 1000 organisms in a particular cross. In this, 250 is going to be having capital Y, 500 is going to have capital Y, small y, and 250 is going to have small y, because we know that 1 by 4th is going to be dominant, 1 by 4th is going to be recessive, and the middle one, that is 1 by 2 or 2 by 4, that is going to be the intermediate condition, that is capital Y, small y. So this is how we are using the ratios to find out the number of organisms which belongs to each of the different genotypes. Here is another example where we have taken tallness of the plant as a characteristic. So in the first generation, that is in the parental generation, we are going to carry out a cross between capital T with small t, that is purely tall with purely dwarf, that is known as pure lines. These are known as pure lines because they are homozygous. Now when the cross is carried out, it results in the formation of only one type of plant, because here it is capital T is the gamete and here small t is the gamete. On fertilization it will form capital T, small t, and this is going to be my F1 generation. Next we are going to carry out a selfing of the F1 generation to yield the F2 generation. So when we are doing the selfing of F1 generation, it is capital T small t with capital T small t. Again we can use the Punnett square. So I'll draw over here. So this is the square, and now we will make the crosses, capital T small t here and capital T small t here. So here it is going to be capital T, capital T, capital T small t, capital T small t, and small t small t. So these are the different genotypes that we are getting. So again, the phenotypic ratio is 3 is to 1 because all these are tall, 1 is dwarf, and the genotypic ratio is coming out to be 1 is to 2 is to 1. So this is a fixed ratio for monohybrid crosses. So moving on with the next topic, that is how exactly is this selfing and crossing carried out. So crossing means we are going to take pollen grains from the anther of one flower and we are going to dust it on the stigma of another flower. So then the process of crossing is done. So this is how the process of crossing is done, and ultimately after the process of sexual reproduction, seeds will be produced, and from the seeds new plants are formed. So it will take a long time. Nearly it will take 3 months minimum for the formation of F1 generation. Though we are writing the complete process in just 5 minutes or 3 minutes, but the process in reality will take more than 3 months, and it depends on the various seasons, on the soil, on the climate, and so on. Now let us talk about the next cross, which is the dihybrid cross. So here I have taken an example here, round yellow seeds with wrinkled green seeds. This is a cross which I am going to do, round yellow seeds with wrinkled green seeds. So I will take the cross one by one. Step by step we are going to solve now. First one is the parental generation. So in the parental generation we are going to take round yellow and wrinkled green. So if I go for the genotype of the parents, we can see that round yellow is capital R, capital R, capital Y, capital Y. And wrinkled green is going to be small r, small y, small y. So here no heterozygotes are there. So the gametes which is produced is capital R, small y. This is the gamete produced. And here it is going to be small r, small y. So in the F1 generation, it is capital R, small r, capital Y, small y. This is the F1 generation. Next, we are going to carry out the process of selfing of the F1 to get F2 generation. So let me take the process of selfing in the next page. Capital R, capital Y, small y is selfed with capital R, capital Y, small y. So now there is a problem. What is the problem? How many different types of gametes will be produced by this and how many will be produced by this? 4, and here also it is 4 because it is heterozygotic condition. So here the gametes are going to be capital R, capital Y, capital R, small r, capital Y, and small r, small y. And here it is going to be capital R, again the same thing, capital R, capital Y, capital R, small r, capital Y, and small r, small y. Now I have to carry out the Punnett square. Now it is going to be a really big job because we have 16 squares. However, we can do it easily. This is how we are making the Punnett square over here, and this is the observation that we are getting over here. But the easy method for getting what are the different phenotypes and genotypes, we can use another method which is known as fork line method. Please do not use this fork line method in the examination because in NCERT fork line method is not mentioned, only Punnett square is mentioned. So you have to proceed with this method for the examination. But if you get any numerical question, especially for the competitive examination, you can use the fork line method. So in fork line method, what are you going to do? You are going to first take these two monohybrid crosses. Whenever you have a dihybrid cross, we are going to assume it as a two monohybrid cross. First one is capital R, capital R, small r, and here we are going to take capital Y, capital Y, and small y. And here we will get capital R, this is capital R, small r, this is capital R, small r, and this is small r. And this is capital Y, capital Y, small y, capital Y, small y, and small y, small y. So these are the different crosses that we are getting. Now with this we have to multiply all the 4. Similarly with this we have to multiply all the 4 in the second Punnett square. With the third one we have to multiply with this, and with the fourth one we have to multiply with this. That's very simple. So let us take the first one, capital R, small r. We have to multiply it with capital Y, capital Y. So it is capital R, small r, capital Y, capital Y. Next is going to be capital R, capital R. This is capital R, capital R, capital Y, small y. Next is also going to be capital R, capital Y, small y. And next is capital R, small y, small y. So like that you can obtain all the 16, and from this you can count the number of different genotypes. And when you are doing that counting, you will be getting the genotypic ratio of the F2 generation as 1 is to 2 is to 1 is to 2 is to 4 is to 2 is to 1 is to 2 is to 1. This is the genotypic ratio, and the phenotypic ratio comes out as 9 is to 3 is to 3 is to 1. So now you will be wondering, how can I remember this long ratio? It is very simple. We have seen that in monohybrid ratio was 1 is to 2 is to 1. I am just taking the square of this. We know that a plus b the whole square is equal to a square plus b square plus 2ab, or it is a square plus 2ab plus b square, right? So here also I'm using the same thing. 3 is to 1 the whole square, that is same as first this one square that I'm going to take here, that is 3 square is to next term is 2ab. This 2ab I'm going to write as ab is to ab. So that is equal to 3 into 1, that is 3, is to again 3 into 1, that is 3, is to this one square, that is 1. So that is how you're getting it as 9 is to 3 is to 3 is to 1. Similarly, when you square this ratio, first of all, what are you going to do? You are going to take this one and then you will find 1 is to 2 is to 1. You will write the same thing. I'll take it here. 1 is to 2 is to 1 the whole square. First three terms are going to be the same, that is 1 is to 2 is to 1. Next three terms are going to be the double of this, that is 2 is to 4 is to 2. And the next three terms are going to be same as the first one, that is 1 is to 2 is to 1. So this is going to be the genotypic ratio of F2 generation for dihybrid cross. So we have seen 4 ratios today. First one was that of monohybrid cross. Genotypic ratio is 1 is to 2 is to 1. Phenotypic ratio is 3 is to 1. And that of dihybrid cross, genotypic ratio is 1 is to 2 is to 1 is to 2 is to 4 is to 2 is to 1 is to 2 is to 1. And that of the phenotypic ratio is 9 is to 3 is to 3 is to 1. Now we are moving on to the last topic of this chapter, which is sex determination. How is sex of a baby determined, whether it is a male or a female? The sperm consists of two types of gametes. One is known as X type of gametes and another is known as Y types of gametes, because the chromosomes are designed in such a way. One of the chromosome looks like this. This is known as the X chromosome, and another will have one small arm. This resembles to the Y chromosome. So this is the X chromosome and this is the Y chromosome. Whereas in the this is seen in the testis. Okay, this is seen in the testis. But in the ovary, you can see only one type of chromosome, that is this. All chromosomes are of the X type. So this is undergoing meiosis, right? Both the cases it is going to undergo meiosis. So only one can get into the gamete. Either it can be X or it can be Y. But here only X can enter into the gamete. So if this X chromosome is going to fuse with the X chromosome, it results in XX condition, and XX condition is going to be a female. And if the Y chromosome is produced by the father and X chromosome produced by the mother, when those two are fertilizing, then it will form XY condition, and XY condition is going to turn into a boy. So who is responsible for sex determination? It is the father who is responsible for the sex determination of a child. So this completes our chapter, that is genetics, heredity and variation, and also the course of biology. We had 4 chapters in biology: life processes, how do organisms reproduce, heredity and variation, and our environment. So I hope you like this lecture, and if you have liked this, please do like below and share and subscribe to the channel so that you does not miss out any new series which will be uploaded in future also. So thanks for watching, and please do share it with your friends. Thank you.